Bismillahirrahmanirrahim. Beta, we are starting to discuss the inflammation. That is the second chapter after cell injury, and this is the series of online lectures for my third year M.B.B.S students. Today in inflammation, we will discuss introduction to the inflammation, the types of inflammation, and the various events that occur during acute inflammation. Basically, inflammation occurs in living tissue, and as you know that we are all living together with microbes, we face daily various types of injuries. So God has gifted us with an immune system that will protect our healthy tissues from these damages that are caused by various types of injuries and by various invading microorganisms like bacteria, viruses, parasites, fungi, and others. So inflammation is basically a protective response. It is a defense mechanism of the living tissue, and the purpose behind the inflammation is that whenever we are attacked by injurious agents like bacteria, viruses, and other microorganisms that we say are the foreign invaders coming from the outside, and the second thing that is the internal trigger for inflammation is the presence of dead tissue. So the purpose of inflammation is to destroy the invading agent, and if cannot do so, then to reduce its toxicity by neutralizing it, by diluting it, and by neutralizing it. So the tissue injury, which is caused by various injurious agents in the form of microorganisms and certain types of physical, chemical, or thermal injuries, they will cause tissue damage, and inflammation will protect the healthy tissues by initiating a response mainly operated by our immune system by destroying, neutralizing, or diluting the invading agent to decrease its toxicity. And also the second trigger for inflammation, which is the presence of dead tissue caused by the inflammatory response or by the injurious agents or by physical or other types of injuries. So inflammation takes the damaged tissue back to a state of health by initiating the process of resolution, which occurs in acute inflammation, and if the injury is mild. However, if the injury is severe, intense, and strong, or if there is some problem when the acute inflammation cannot resolve the response, then we proceed to the chronic inflammation, and chronic inflammation mostly result in healing and repair. So basically what we learn that inflammation is a defensive host response to foreign invaders and necrotic tissue. Remember these two triggers. The inflammation is intended to eliminate the initial cause of cell injury, as well as the dead and damaged necrotic tissue and cells that may result from the original insult. And in addition, inflammation will initiate the process of restoring the damaged tissue to a state of healthy and normal tissue. But if it can't do so, as occurs in chronic inflammation, then inflammation will start the process of repair. In repair, the dead or damaged tissue is replaced by the scar tissue, which is physiologically not as functional as the original tissue, but anatomically it restores the integrity of the tissue. Unfortunately, inflammation causes the effects that I discussed by destroying, diluting, or neutralizing the invading agents, but potentially inflammation along with the damaged tissue can damage the healthy body tissues. If inflammation is limited, controlled, and if it is of short duration, it will play its beneficial role. But if it is prolonged, then there is a risk of causing damage to the healthy tissue. And this inflammation induced damage is the target of many pharmaceutical and clinical diseases and their treatment. We divide inflammation into two types. If it is of short duration, which mostly lasts for less than 4 weeks, we say this is the acute inflammation. And acute inflammation potentially can result in resolution and restoration of the damaged tissue to a healthy tissue and a fully physiologically functional tissue. However, this depends on the severity and intensity of the invading agent. Automatically, if the invading agent is severe, if it is prolonged, or if there is some problem in our immune system, for example, if a person is immunocompromised, and if the inflammation in its acute form cannot resolve the problem, then it will evolve into chronic inflammation. So clinically, if inflammation lasts for more than 6 weeks, we say this is chronic inflammation. In between the two, there is a third type which is known as subacute inflammation, and we say it if it is more than 4 weeks but less than 6 weeks. At that stage, we say this is subacute inflammation. As I said, that inflammation is intended for its beneficial role, but in that situation, the inflammation, if it is short-lived and if it is self-limited, and if the person is immunocompetent, healthy, so inflammation will play only its beneficial role, and it will make the tissue to restore to its normal physiological and anatomical function that we say is the resolution. But in conditions when the invading agent is strong, for example, when there is heavy infection or infection with highly pathogenic organisms. Secondly, if the inflammation or the inflammatory process is prolonged. And when it will occur, when the invading agent resists the inflammatory process and cannot be easily eradicated. So in that situation, the inflammation carry high risk of damaging the healthy tissues. And a third condition is that when the inflammation is inappropriately directed. In that situation, we get two pathological conditions. One is the autoimmunity. In autoimmunity, what happens that normally our immune system recognize self and non-self invading agents. And in that way, our immune system normally doesn't develop any inflammatory response against the tissues or of our own body. But there are certain situations in which the immune cells fail to recognize the body's own tissues and proteins and develop an inflammatory response against the body's own tissues and result in a large area of diseases known as the autoimmune diseases. So in that situation, we say that the immune response is inappropriately directed. And there is another condition that we discuss in immunological disorders, and we say that is simply allergy or hypersensitivity. By allergy or hypersensitivity, we mean that there are so many allergens and antigens present, but normal people do not develop an abnormal inflammatory response to them. For example, in asthmatic patients, but certain individuals, they are genetically susceptible. There is some problems in their genetic makeup and they develop an unnecessary, exaggerated and highly strong response known as the allergy or hypersensitivity. So in these situations, like as I said, in autoimmune disorders, in autoimmune diseases and allergic or hypersensitivity disorders, the inflammation is inappropriately or exaggerated response occurs, and automatically these two conditions will lead damage to the normal tissues. In these conditions, if the inflammatory process is prolonged for more than 6 weeks, or due to some any reason it is poorly controlled, so it will lead to chronic inflammation. The differences, 4 major differences between acute and chronic inflammation are that in acute inflammation, the onset is fast. Usually it takes minutes or hours. In comparison, in chronic inflammation, the onset is slow. It may take days and months. The immune cells in acute inflammation are mostly the neutrophils, which is the first defense line against the invading microorganisms, but they are not so strong, and if the problem remains, they are replaced in 24 to 48 hours by mononuclear cells, which are supposed to be more stronger, and they are the monocytes that are converted into macrophages in the tissues, and the lymphocytes. The lymphocytes are part of the acquired immune system. Now coming to the outcomes, tissue injury and fibrosis. In acute inflammation, it is usually mild and self-limited. But as the inflammation persists longer and longer, it carries the risk of severe and progressive tissue injury, and automatically the problem will lead to fibrosis and scarring in the tissues. Now, coming to the clinical signs and symptoms, in acute inflammation, they are very prominent. Pain, fever, they are very prominent, but they are less prominently experienced by patients when it is in its chronic form, and sometimes before the patient develops signs and symptoms, lot of time is elapsed. Now, inflammation is a multi-step process, and we must remember the various steps in inflammation by the remembering the coin, 5 rupees coin. 5 are, first thing is that our immune system should recognize the injurious agent, that it is foreign and it is injurious and it is damaging to the body. Right? Now, after recognition of the injurious agent, then the body prepares and carries its army in the form of leukocytes to fight against the invading agent, and that is the second step known as the recruitment of leukocytes. Now these leukocytes will destroy, they will neutralize, they will dilute accordingly, and will attempt to remove the offending agent and to finish the initiating cause. And the fourth step is the regulation of the response. The inflammatory response must be appropriately regulated, because if it is left unregulated, it will cause damage not only to the invading agent but also to the healthy body tissues. And at the end, the fifth R is the resolution or repair. In acute inflammation, mostly resolution occurs, but in chronic inflammation, repair and scarring of the tissue is more prominent. The initial events when an external or internal triggers initiate the process of inflammation will cause two changes: vascular changes, by that we mean changes in the blood vessels and blood circulation, and cellular response, by that we mean to carry the defense and cells and leukocytes and to recruit them, to activate them, and so that they are fully functional to respond to the invading agent. The 5 cardinal signs are known as the heat, calor, redness, rubor, pain, dolor, swelling, tumor, and finally the loss of function. These are the cardinal signs of inflammation, and they are mostly asked in exams. Heat, whenever the blood supply to a tissue is increased, automatically it will produce warmth in that area. Secondly, the area will become red, and during inflammation, various cytokines that are released during the process, and moreover stretching of the sensory nerve endings, it will cause pain. The swelling or tumor, it will result that when during the process of inflammation, fluid leaves the blood and goes into the extravascular site, so accumulation of fluid or exudate during inflammation, it will result in swelling, and automatically that part of the body will be unable to function normally, at least for some time. Now, what causes inflammation? Number one, the infections by microorganisms, and we know that the microorganisms are bacteria, viruses, fungi, parasites, and rickettsia. The second cause of inflammation is in the form of physical injury, like trauma, extremes of temperature, and radiation, which is very harmful and damaging, and the various chemicals present in the environment, particularly in the industrialized areas, and sometimes foreign body, like a bullet. So the third type which causes inflammation is the dead tissue necrosis and ischemia, when the tissue is deprived of its blood supply. So ischemia is a common cause for the initiation of the inflammatory process, and as do so the necrotic or dead tissue. And as we said that immune reactions, inappropriately directed immune reactions in the form of hypersensitivity or autoimmunity, so all these causes will lead to acute inflammation, and the acute inflammation will attempt to remove the cause, to remove the dead tissue, to start the process of resolution. But due to any reason, for example, if the injurious agent is stronger, if it is persistent, and if it resists removal by the acute inflammation, so what happens? The acute inflammation will evolve into chronic inflammation. As we discussed that the two triggers for inflammation, we divide them into external triggers and internal triggers. We divide the external triggers into those that occurs due to microorganisms, bacteria, viruses, fungi, parasites, and those due to non-microbial. So the first thing is, I said that our immune system, our body, our inflammatory cells, they must recognize these external factors. And the microorganisms like bacteria, they have two types of I can say weapons. One includes the virulence factors that give support and help the invading microorganism to cause damage. And fortunately, these microorganisms have certain red flags which are known as the pathogen associated molecular patterns, or PAMPs, and our inflammatory cells recognize these PAMPs. If they can't do so, inflammatory reaction cannot be initiated. So the PAMs, pathogen associated molecular patterns, are certain components in the microorganisms that are recognized by our immune cells. The external triggers also include certain allergens, irritants, and toxic compounds that we say are the non-microbial. So the external factors or the external triggers are recognized by the immune cells through the recognition of the PAMs, pathogen associated molecular patterns. The internal triggers for inflammation, they are recognized by the immune cells through the DAMPs, damage associated molecular patterns. And damage associated molecular patterns are red flags with the dead cells, and if the plasma membrane of the cell is injured, our immune system recognize, and these are known as the damage associated molecular patterns, and they constitute the internal triggers. Now what will happen, that if the immune system is stimulated after it recognizes the external factors or internal factors, it will initiate the inflammatory response. And the purpose of the inflammatory response are the goal will be to eliminate the cause, to remove the dead and necrotic cells, and to start the process of tissue repair. As I said that the microorganisms, they contain two things. They have virulence factors that help them to colonize tissues, to cause infection, and to override our immune system. So they are supporting the microorganisms to make infection and cause injury. But these microorganisms, they have certain components which are known as the pathogen associated molecular patterns. And these pathogen associated molecular patterns, they are in the form of red flags. Our immune system, our inflammatory cells recognize these PAMs. In bacteria, the pathogen associated molecular patterns include some conserved patterns in their cell membranes, like polysaccharides, peptidoglycan, lipopolysaccharides, lipoteichoic acid, and mannan in the fungi, while the viruses, they do not have a cell membrane. बेटा वायरस और बैक्टीरिया में मेजर डिफरेंस ये है कि बैक्टीरिया में डीएनए और आरएनए दोनों होती है. Therefore, it is capable of enjoying or surviving an individual life. But वायरस में या डीएनए होती है या उसमें सिर्फ आरएनए होती है. So independently they cannot survive, and for their survival the virus will use the live cell, body cell. It will enter into the cell, and in the nucleus of the cell it will use the DNA or RNA of that particular cell to live and to make replication and to divide. So the pathogen associated molecular patterns in the case of bacteria are mostly the cell membrane components. But since the viruses do not have a cell membrane components, so mostly they are recognized by the viral RNA or DNA which is present in the healthy cells. And these components constitute the pathogen associated molecular patterns. So now we proceed. What happens that our immune cells recognize the external factors and the internal factors. They recognize the external factors through pathogen associated molecular patterns, and they recognize the dead tissue or internal factors through damage associated molecular patterns. And the inflammatory cells have receptors for the recognition of these PAMs and DAMs, and these receptors are known as the pathogen recognition receptors. And once the pathogen recognition receptors recognize the PAM or DAM, so the interaction of the PAMs and DAMs with the pathogen recognition receptors on the immune cells will then initiate the process of inflammation. The major inflammatory cells, we divide them into granulocytes, those cells that have granules in their cytoplasm, for example neutrophils, eosinophils, and basophils, and the mast cells, and into agranulocytes that are lymphocytes and monocytes. Monocytes normally circulate in the bloodstream, but when they are needed, they leave the bloodstream, go into the extravascular tissue, and are present in the form of macrophages and dendritic cells. Macrophages are derived from the monocytes, as the dendritic cells known is the antigen presenting cells, and these are more strong and efficient in killing the microbes as compared to the monocytes. So we conclude that these inflammatory cells recognize the external factors or microorganisms through the pathogen associated molecular patterns, and they recognize the internal factors from the dead tissue or necrotic tissue through damage associated molecular patterns, and these two, the B.A.M.S and D.A.M.S, interact with the pathogen recognition receptors present in the inflammatory cells to initiate the process of inflammation. Once the cells, inflammatory cells, recognize the B.A.M.S and D.A.M.S, they release cytokines, and these cytokines, for example, the histamine, which is present in large amounts in the mast cells, these cytokines, they will act as mediators of inflammation. They are the players of inflammation, and for example, the histamine released by the mast cells will cause the two initial events that occur during inflammation, acute inflammation. That is the vasodilation, to increase the caliber of the blood vessels to carry more blood and cells to the area of injury and inflammation, and to increase the permeability. By that we mean to make the vessels more permeable so that the cells, they leave the blood and go into the site of extravascular site where the infection or injury has occurred. And the cytokines released by various inflammatory cells will activate the endothelial cells of the blood vessels, and we discuss in the next slide that how the leukocytes get adhered and how do they migrate through the blood vessel wall into the extravascular tissue. And here important thing one is that this is not only the cells of the immune system or the mast cells, but also the endothelial cells, when activated by cytokines at the site of injury or inflammation, they release nitric oxide, which itself is a vasodilator. So remember the two components: vascular component, which consists of vasodilation, increasing the caliber of the blood vessels with increased blood supply and carrying more and more inflammatory cells to the area of inflammation. And the second thing, the second component of the vascular changes is the increased permeability. Basically, in inflammation, two things we discuss: vascular changes and the cellular changes. So the two major vascular changes are the vasodilation and increased permeability, for the inflammatory cells to leave the blood and to go into the extravascular site where the invading agent or the dead tissue is lying. But before we proceed to discuss the increased permeability that occur during inflammation, let us come to the normal physiological state of a capillary. You know that the capillary is a junction between the arterial and the venous system. One end of the capillary is at the end of the artery, and the other end of the capillary is at the beginning of the vein. So this is the part that communicates the arterial and venous blood vessels. Normally, the blood pressure which provides the hydrostatic pressure at the arteriolar end of the capillary, it will cause fluid to leave the blood vessels and go into the extravascular space. But To minimize this leakage or this outpouring of the fluid from the blood into the extravascular space, or blood contains the plasma proteins mostly synthesized by the liver, and these proteins provide the oncotic pressure, colloidal osmotic pressure, and this colloidal osmotic pressure will pull the fluid back into the blood. At the arteriolar end, the increased hydrostatic pressure causes fluid to go into the extravascular space. By doing so, the blood becomes concentrated, more hyperviscous, and as it reaches at the venular end of the capillary, the plasma colloidal osmotic pressure, it will balance the hydrostatic pressure, it will be more than hydrostatic pressure, and it will pull the fluid back into the blood. Some of the fluid that remains in the extravascular tissue is drained by the lymphatic system. So we divide this accumulation of extravascular fluid, in which the fluid leaves the blood and goes through the vessel wall, through the endothelium into the extravascular space, into transudate and exudate. Transudate is a thin fluid that will go due to increased hydrostatic pressure into the extravascular space, or due to decreased plasma colloidal osmotic pressure. Now, any disease or any condition that cause increase in the hydrostatic pressure, for example in congestive heart failure when there is the venous outflow obstruction, or any condition in which the amount of the plasma proteins is decreased, either due to decreased formation or due to loss from the body, as occurs in nephrotic syndrome and other kidney diseases. So when the colloidal osmotic pressure or the oncotic pressure is decreased, automatically the increased hydrostatic pressure or the decreased plasma colloidal osmotic pressure will cause fluid to accumulate in the extravascular tissue. The third cause can be lymphatic obstruction due to any reason, tumor, inflammation, because if the lymphatic system fails to clear the fluid from the extravascular tissue, the fluid will get accumulating there and will cause the formation of edema. But remember that this fluid that we say transudate, it has very small amounts of proteins and very small amounts of cells, because the endothelial wall of the blood vessel is not so much permeable. The gaps are not so much open to allow cells and proteins to leave the blood and to go into the extravascular space. So the transudate will be a low protein and containing a small amount or a few cells. So we have understood the transudate. Now let us come to inflammation. In inflammation, what happens that the game is not played by the hydrostatic pressure or the colloidal osmotic pressure totally. Here what happens that the gaps in between the endothelial cells are increased and the blood vessels in the area of inflammation are turned open as compared to the edema formation that occurs in transudate, and only thin fluid is allowed to escape into the extravascular tissue. During inflammation, what happens that the increased permeability of the blood vessel allows the proteins, mainly fibrinogen, and as well as the cells to go from the blood into the extravascular space, and the fluid accumulated as a result of this increased vascular permeability is known as the exudate. It is exudate, not transudate, because it contains a high cellular content and high protein content. So what we said, we summarized that is summarized in the book. Inflammation is a defensive host response to foreign invaders and necrotic tissue. Remember these two things. They are triggers from inflammation: invading agents and dead tissue. The main components of inflammation are vascular events and cellular response. And these two responses are activated by inflammatory mediators, and the chemical mediators of inflammation that we discussed in our next video, they are either derived from the cells in the particularly in the localized area of the inflammatory reaction, or they are derived from the circulating precursors that are mostly produced in the liver and circulate in the blood in inactive form, but when needed they are activated by their enzymatic breakdown. And these plasma derived factors and cell derived factors, they make the chemical mediators of inflammation, and these are the chemical mediators of inflammation that are players of the match. And any problem is if any chemical mediator is not properly functioning, automatically you can say that if we miss a player in a match, the match will not be played efficiently and effectively. Right? Now, as I said, inflammatory response is a multi-step process, and we start from the recognition of the injurious agent by the immune cells, then by inducing the vascular changes, bringing more and more cells to the area of damage by increasing the blood flow. As a result of vasodilation, the inflammatory cells or leukocytes are recruited to that area. Now they will fight against the invading agent and will remove it, and the inflammatory reaction should be very appropriately controlled, because if it is not controlled, it will damage the healthy tissue, as inflammation potentially can cause damage not only to the invading agents but also the healthy tissues. And the final step will be to finish the process, to resolve the process. And mostly in acute inflammation, if the injurious agent is not so severe, not so strong, and if it is not so persistent, it will result in resolution. While in other case, if the primary inflammatory response fails to resolve, it will evolve into chronic inflammation, and chronic inflammation mostly results in extensive tissue damage that we say repair. By repair we mean that the original anatomical and physiological integrity of the tissue is not maintained, but just the damaged area, the damaged tissue is replaced by scar tissue or fibrous tissue. जिस तरह कपड़े में बस सिर्फ एक पेवन लगाते हैं हम. The outcome of acute inflammation will be either elimination of this invading agent, followed by decline of the reaction and repair of the damaged tissue, or if the acute inflammation fails to resolve the problem due to any reason that we discuss further, it will lead to chronic inflammation. The vascular changes and the cellular elements are the two components of acute inflammation, the initial components, and the purpose of the vascular changes is to ensure a quick delivery of the leukocytes and plasma proteins uh, to fight against the invading agent or the dead tissue. And the vascular changes are: number one, vasodilation, increase in the size of the increase in the caliber of the blood vessels to carry more blood, and the second is to make the blood vessels porous, permeable, so that inflammatory cells and proteins can leave the blood and can go into the site of the damage. During this process, the endothelial cells are activated by various cytokines, and also the inflammatory cells are activated so that they become adhere to the endothelial wall, and there is the possibility that they should migrate through the endothelial wall into the extravascular space, that we discuss in detail in next. And once the vascular changes has occurred, vasodilation has occurred, there is increased permeability, what will happen? That then we proceed to the cellular events, and the cellular events will be to bring more polymorphonuclear leukocytes to the site of injury after the vascular changes in the form of vasodilation and increased permeability. What happens that the neutrophils are polymorphonuclear leukocytes in acute inflammation, and the mononuclear cells are chronic inflammatory cells later on, most commonly during the chronic inflammation. So they are recruited to the area, and after the cells, inflammatory cells, are recruited to the area of inflammatory reaction, now the next step is that they should be activated, because without activation they will be unable to perform their functions, that is to fight against the invading microorganisms. And the recruitment and activation of the inflammatory cell is performed by various chemical mediators of inflammation that we discuss next. So here two changes occur. One is the vasodilation and the second is the increased permeability, and the purpose behind these two events is to recruit leukocytes, to bring lot of leukocytes to the area of damage or to the area of inflammatory reaction. And this is achieved by first, whenever there is body is invaded by any external trigger, what happens then, that we study in hemodynamics, when we study the normal hemostasis. Initially, due to nerve stimulation, there is a brief transient vasoconstriction, and the purpose behind this transient vasoconstriction is to decrease the blood flow, and if there is a cut or if the area is injured, to reduce the blood loss. But this is a very transient response, and it is followed by the arteriolar vasodilation, and the purpose behind the arteriolar vasodilation is to bring and recruit inflammatory cells and proteins, and this vasodilation is responsible for the two cardinal signs in the form of erythema and warmth. The second thing that happens is the increased permeability, and due to increased permeability, when the blood vessels become more porous and permeable, the protein rich fluid accumulates in the extravascular tissue and develop into edema that we call as the exudate. Now what happens, that when the fluid leaves the blood becomes more concentrated and it becomes more hyperviscous. Normally, the flow of the blood in a vessel, like water flowing in a pipe, or we can say in a river, that is called as the laminar flow or axial flow. And in laminar flow or axial flow, the blood cells, they flow in the central zone or the speed zone, and the plasma, it flows in the peripheral zone. But as during inflammation due to increased concentration of the blood, when the fluid leaves the blood and the blood becomes more viscous and concentrated, there is stasis. The flow of blood is decreased, the speed of the blood is decreased, and this stasis will disrupt the axial or the laminar flow, and the cells that were flowing in the central speed zone, they will get to go into the peripheral zone, and this is known as the margination, and that is an important step in the vascular changes that occur. Now, for the cells to leave the blood and to go into the extravascular site, what happens that the endothelial cell, they contract, and this contraction is brought about by certain inflammatory mediators and cytokines, and when the endothelial cells contract, they produce gaps in between the cells, particularly at the post capillary venules. Moreover, the initial injury or later on the injury induced by the leukocytes will cause damage and necrosis and detachment of the inflammatory endothelial cells, forming holes in the blood vessel. And the third component that account for the accumulation of fluid and proteins in the extravascular tissue during the process of inflammation is the transcytosis of proteins. In this way, in this situation, the fluid doesn't go through the interendothelial gaps, but they go through intraendothelial process, entering the endothelial cells from the blood and leaving the endothelial cells on the extravascular side. So this is called the transcytosis of proteins, and this also accounts to the increased accumulation of exudate and fluid rich in proteins and cells. And as during inflammation, we will discuss further, a process occurs particularly during chronic inflammation that is known as the angiogenesis. And in the angiogenesis, what happens that new blood vessels are formed to supply the damaged area, or in chronic inflammation the fibrous tissue, scar tissue, and these newly formed blood vessels, they are very fragile, and fluid can easily leak from these blood vessels, and up to certain amount they put their input and account for the increased vascular permeability and accumulation of the fluid in the extravascular tissue. So here we summarize the vascular reaction in acute inflammation is that the vasodilation, which is caused by the various chemical mediators, particularly the histamine, which is released from the basophils and mast cells, and this vasodilation is responsible for the cardinal signs of redness and warmth. And during the vasodilation, the blood flow in a vessel of higher caliber than the normal one, and automatically what happens that the speed of the blood is decreased. On the other hand, when there is increased permeability, which may also be induced by histamines, kinins, and other chemical mediators, they will produce gaps in between the endothelial cells, and these gaps are also produced by injury due to the initial cause to the area of to the blood vessels in the area of the injury or inflammation, and sometimes the leukocytes, as we discuss further, they can also cause endothelial injury, because leukocyte, the main purpose of the leukocyte is to cause damage to the microorganisms, but as a side effect they have the potential to damage the healthy tissue, and they can also cause injury to the endothelial cells, and this will support the increased permeability and passage of fluids through the endothelium. Automatically, as a result, the increased permeability will lead to accumulation of proteins and leukocytes to leave the blood and to go outside the vessel wall into the area of infection or inflammation, and they will cause accumulation of fluid in the form of exudate or edema. Now, here again we summarize the sequence of events: recognition of the offending agent, which is mainly done by the antigen presenting cells like macrophages, dendritic cells, and others, and they will lead to the vascular changes in the form of vasodilation and increased permeability. And then the role of the cellular element of the inflammation, and that is the migration and recruitment of leukocytes to the site of injury. As I told you that the increased blood flow carries more and more polymarbonuclear leukocytes to the site of blood injury to fight against the invading agent. Now, these phagocytes will kill and degrade the offending agent, for example, a bacterium or a virus, and this they do so by a process known as the phagocytosis. They engulf the invading agent. They, for example, they engulf the bacterium, and inside these inflammatory cells in their cytoplasm, they kill these organisms, microorganisms, by the process of phagocytosis that we discuss in detail next. And the final outcome in acute inflammation and in a person with a good health and immunocompetent, with a healthy immune system, it will result in resolution. And if the person is immunocompromised or there is some problem in the inflammatory response, for example, if the invading agent is very resistant to be killed by the leukocytes or inflammatory cells, or if it is inherently damaging the cells, so the problem will here evolve. The acute inflammation will evolve into chronic inflammation, and chronic inflammation always cause extensive tissue destruction that will result in fibrosis and scarring. Now, leukocyte recruitment. Leukocyte recruitment is a very important step. As I told you that normally the leukocytes, they flow in the speed zone inside the blood vessel. But during vasodilation and during increased permeability, when the speed of the blood flowing through the blood vessel, when it is decreased, the leukocytes, they leave the central speed zone and come to flow in the peripheral zone of the blood vessel, and this we say is margination and rolling. Here what happens, that certain molecules known as the selectins. They are expressed on the endothelial cells, and selectin molecules that like weak glue. And what happens that initially the leukocyte attaches and detaches, attaches and detaches. This is known as the rolling. After the margination and rolling, then firm attachment of the leukocyte to the endothelial wall becomes important, and this is achieved by the molecules known as the integrins. एंड इंटीग्रीन्स जो है ना उसका मिसाल ये है कि वो एक किस्म का स्ट्रांग ग्लू है. वंस ल्यूकोसाइट्स आर एक्टिवेटेड बाय द इंटीग्रीन्स, they are firmly attached to the endothelial wall. So the initial event was margination, coming of the leukocytes to the peripheral zone, rolling, attach and detach, attachment and detachment, then the expression of the integrins which cause firm adhesion of the leukocytes to the endothelial blood vessel wall, and then through the increased gaps in between the endothelial cells that we discussed, what will happen that the leukocytes, they will leave the blood and go into the extravascular site to perform their major function. And this is known as the transmigration between endothelial cells, and the leukocytes move just like M.B.B.S Students move by pulling out the pseudopodia, and this is we can say amoeboid movement. Now once the leukocytes are outside the blood vessel in the area of the tissue injury, then the inflammatory cells in those area release certain cytokines known as the chemokines that we discuss in detail later during the inflammatory mediators, and the same are produced by certain microorganisms, and they act like a magnet. They will attract the leukocytes, the recruited leukocytes, to the area of the tissue injury, and the inflammatory cells, for example the macrophage, when they ingest the microbe initially and function as the antigen presenting cells, they will release cytokines and other chemical mediators, for example selectin, which will cause rolling and loose attachment of the leukocytes to the endothelial wall, and then integrins that will cause firm attachment of the leukocytes to the endothelial wall, and also certain chemokines that will attract and cause directional movement of the inflammatory cells to the area known as the chemotoxins. So leukocyte recruitment, briefly it is margination and rolling along the blood vessel wall, which is caused by the selectins, firm attachment which is caused by the integrins, and then movement of the leukocytes through diapedesis, amoeboid movement, and this will cause the leukocytes to leave the blood and to go into the extravascular tissue. And here certain cytokines known as the chemokines that act as magnets and induce directional movement of the leukocytes to the area of damage, and migration supported by the chemotoxins, which is done by certain chemotactic stimuli and certain chemoattractants which are released by the inflammatory cells and also by certain microorganisms, and the migration of the inflammatory cells to the interstitial tissue. हमने आज क्या पढ़ा इसमें? ल्यूकोसाइट्स आर रिक्रूटेड फ्रॉम द ब्लड इनटू द एक्स्ट्रावेस्कुलर टिश्यू, जहाँ पे इन्फेक्शियस पैथोजेंस आर डेड टिश्यू जो है ना, इट इज लाइंग, एंड द purpose of this recruitment and activation is to remove, neutralize, detoxify the invading agent. And we discussed that recruitment of the leukocytes is a multi-step process. Initially, the loose connection to the endothelial wall known as the attachment and rolling, and which is mediated by selectins, and then this is followed by the firm attachment which is mediated by integrins, and finally the migration of the leukocytes through intraendothelial junctions into the extravascular space. Now, the chemical mediators of inflammation include various cytokines which promote expression of the selectins and integrins that are responsible for the margination, rolling, and firm attachment of the leukocytes to the blood vessel wall or endothelial wall. And for example, the tumor necrotic factor and interleukin 1 and some cytokines, they act as chemokines, causing a directional movement of the leukocytes towards the area of injury and inflammation. And these inflammatory mediators, along with other cells, they are produced by the tissue macrophages and other inflammatory cells that respond to the pathogens. And we said that in acute inflammation, the predominant cells, the initial first line cells attacking the invading agent are the dead tissue. They are neutrophils, but later on they are replaced by mononuclear cells and lymphocytes, monocytes and macrophages. Thank you very much.